Here we go. So today we're happy to welcome Henry Abarbanel from the Department of Physics at UCSD and the Scripps Institution of Oceanography. He is a physicist. He got his bachelor in physics from Caltech and the PhD in physics from Princeton. And he has been interested among many other things, but like also like in neuroscience, like data, like forecasting in neuroscience and modeling in computational neuroscience. And today is going to talk about a new method devised to predict the voltage membrane of models of neurons and also like in biological neurons. So without further ado, like Henry, please.
Okay, excuse me. Thank you all. This is the title of the talk, but I want to tell you what it's going to be about. I've been interested in a way to build predictive models of neurons from data alone. What I'm really interested in is building networks of neurons. And I realized that if you have to put at every node of the network, at the neurons, complicated Hodgkin-Huxley models, which you can learn from biological experiments, and I'll show you some of the experiments today, that the computational load is really enormous. And as you grow the number of neurons in your model, if you want to keep biological information, you simply have to simplify the model. And I'm going to show you how we do it from current clamp experiments. One of the people attending is Dan Melisa, now at the University of Virginia, who did these experiments, and I'm glad to have him here. So let me describe that. All right, this is what we do in neurobiology. We have experiments. We have a stimulating current that drives the dynamics of a neuron. This is the shorthand notation for the Hodgkin-Huxley model. And here is the longhand notation, I guess one would call it, for the simplest Hodgkin-Huxley model, which has sodium, potassium, and channel. Here's the leak current, and then additive to that, and that will be important in what we do, is the stimulating current. And I will make a comment later that it is this kind of equation throughout the physics that I know that the stimulating or forcing is additive, and there's a good reason for it too. I'm going to talk about this particular Hodgkin-Huxley model as an example before I talk about what we do with laboratory data, just to show you, to illustrate the idea. And so the question we're asking is, if you know that the state variables, in this case of this simple neuron, are four-dimensional, the membrane voltage, the two sodium gating variables, and potassium gating variables, but we can only measure the voltage. There are no gating variable ohmmeters that I know of as of today. Can we build a model from the data alone that predicts the voltage? And why do we want to do that? So here's what I said in the introduction. We want to build networks, and we want to build big networks so that we can actually represent the biological background of the functional networks we're interested in. I'm going to talk about a specific functional network, which is that of the birdsong system, and in particular the zebrafinch biological network. This network, which is small on biological scales, it's only few times 10 to the fifth or maybe 10 to the sixth neurons as a whole, does in the zebrafinch one thing. The zebrafinch learns and produces communication, it produces vocalization, and it sings the same song as an adult throughout its adult life after a learning period where it learns it from other birds in its group. I'm going to remind you now that the voltage is the only neuron state variable that we need for neurons to communicate with each other. And here is the graphic for that. Suppose you have ligand-gated interactions, so synaptic interactions that have a gating variable which tells you how, when an action potential comes down, this red thing's supposed to be an axon, comes down to near the postsynaptic cell, there is a gating variable that tells you how the neurotransmitter that crosses the synaptic cleft docks and then undocks from the postsynaptic cell, causing currents to flow in. And those currents are a variety of ions. Anyway, there's a time constant associated with that. And the synaptic current involves Ohm's law. This is a conductance. This is the other part of Ohm's law. There's a reversal current, and it involves only the postsynaptic potential here. And there's a gating variable here, I call it S, that depends on the presynaptic potential. But it doesn't depend on all of the gating variables and all of the complicated stuff that's going on in the actual presynaptic or postsynaptic neuron. And so it would be nice to have a reduced model that only knows about the voltage, can't tell you about the gating variables. It doesn't know about them because they're not observed. And that's what I'm going to talk about today. If you have a gap junction connection, which is simpler than the synaptic connection, then the current is just a maximal conductance times the difference between the presynaptic and postsynaptic variables. So the same thing is true there. All right, and this is just a reminder of what a synaptic connection looks like. The differential equation for the synaptic gating variable is pretty familiar. It's a first order kinetic equation, and there are two time constants with this. When the presynaptic voltage is basically off, there's no spike coming down the axon, then this quantity I call S0, which I take as an example to be a hyperbolic tangent, is effectively zero, and ds dt is effectively minus s over a time constant, and the current decays away to zero, meaning nothing's happening there. When the presynaptic voltage rises, because an action potential arises at the synaptic terminal, the S effectively goes to 1, and ds dt is 1 minus s over a different time constant, and that tells you the rate at which the neurotransmitter is bound to the postsynaptic cell. So this is a simple description of it, but again, let me emphasize that the only thing you need are the voltages. Okay. And so what is the bottom line? What I will get to after I show you how this general construction works and show you that it works on experimental data, is that if you only have to predict or forecast the voltage from a given time to the next time over whatever delta t you choose, you can reduce the computation by, in our calculations, we've shown that it's at least the order of a factor of 10 or more times at every node, every neuron in the network. That means that we can build bigger and faster networks than we could without this device. Okay. Just to remind you, there are other reduced dimension neuron models. Integrate and fire models are a class of them, but the structure of these models is typically not based on biophysical experiments. It's based on some good assumptions and some good ideas, but you have to make a number of assumptions about the details even of these integrate and fire models. We don't have to do that, as you'll see. All right, another comment, which I won't talk about in detail today, but let's remember that biological networks typically have many different kinds of constituent neurons. In the songbird example, in the the network that acts as the interface between the sensory, the auditory system, and the song production, there are perhaps 50,000 neurons, and they come in three kinds. One is an interneuron, and the other two are projection neurons to other neural circuits in the avian brain. And so in principle, and that's what our goal here is, you can characterize each of those three classes of neurons, build what we call a DDF, which is data-driven forecasting neuron for each kind, and build a network which you can then test against experimental protocols on the network as a whole. That's the goal. Here's how we do it. All right, I said that. All right, I'm going to begin with a more general formulation than just neuroscience. And as I say that, it will probably occur to you other applications, but this one was really right there on the front burner because we have a wonderful collection of data on which to test the idea. So here's the notation. Whatever your dynamical system is, whether it's a neurodynamical system, a geophysical system, whatever it is, if you can observe D measurements at every time, and so here time is in steps of, we call it H, which is delta T, if you wish. You can observe data over this period of time. If we knew the dynamical rules that took us from time n to time n plus 1, we would build a model, a Hodgkin-Huxley model. But we're going to ask, what is the situation when we don't know the rules? So here is the rules if in a general format. This is a discrete time map from time t sub n to time t sub n plus 1. That's the linear part. And there's a nonlinear function, which includes the details of a Hodgkin-Huxley model or whatever. But we don't know that. The name given to this function in the dynamical systems literature is called the vector field. And we don't know that. And we have to somehow learn properties of this from the data that you collected. And so here's the technical issue that we have to face. We don't know this vector field. And so we have to have a way of representing F in a kind of universal way. And there is such a way that was explored extensively by applied mathematicians and physicists long ago in the 1980s and 1990s. It's quite refined, and I will describe what it is. And here's what how to think about it. This function that we don't know is a function of variables in d-dimensional space. The data is a selection of points in that d-dimensional space labeled by time, and it provides an orbit It could be a strange attractor, or it could be a regular orbit, depends upon the details of the way that the neuron is driven. But we're sampling a distribution of points in that space. What we want is a function of the state variables and some parameters. I'll tell you about the parameters as we go along in detail. If we, a function that interpolates among the points that are observed. If we knew that, we would then have a function in the appropriate space of observables, and we could then use that function for doing prediction. Here it is, prediction component by component of the observed variables with the representation of this function evaluated at time n. So this gives us a discrete time map that tells us beyond our observation window, where we collect the data, we can predict what's observed. To come back to neurodynamics, since we don't observe the gating variables, we can't predict them using the tools that I'm about to describe for you. But that's why I started by talking about what we actually need, which is the voltage in a network. We do not need the various other biologically interesting aspects of every individual neuron. All right, the method that we use to represent the vector field is called radial basis functions. Some of you may know about that, others of you may not. They are functions, and I'll describe examples of them, in which you take your data, you choose a subset of the data, which is the sampling of the distribution of points in your observation space, d-dimensional here, and you make a linear combination of these basis functions the same way as you would with any functions on U. They could be polynomials, they could be whatever, and I'll give you some examples of these functions. You select out of all of the data points that you have seen something that are called centers, and I just said that we have n sub c for centers, and there's a parameter in all of these functions which tells you over what distance in that u-dimensional space this function u is sensitive to the centers that are nearby. So it's a relatively straightforward idea, and the fact that it's a linear combination means that the parameters we have to learn, the parameters are the weights of the particular functions. They're called radial basis functions because this only depends upon the square of the distance between where you are and those sampled parts of the distribution, the centers. So the parameters in here are this width and the weights, which we're going to determine. Frederick, it's all right with me if people ask questions. I don't mind being interrupted if there's any questions to go along.
Yeah, please just ask questions, as don't need to
Okay.
raise your hand or anything else.
All right, I don't know that I'll see the hand raised, but it's okay. Just speak up. Okay.
Question?
Oh, yeah.
Yeah. So is the dimensionality an important aspect of this?
Is what?
The dimensionality.
With dimension of what? Of the measured quantities?
Yes.
No, that's entirely up to you experimentally.
Okay.
In the neuroscience applications, which I'm going to focus on, as I said, because there's data to test the idea on, there's only one thing you can measure, which is the voltage. So the dimension is 1.
Yeah, but I mean, if you can measure the voltage of many neurons at the same time.
No, the idea is that you do current clamp experiments on individual neurons.
Okay.
You measure the voltage of each one, and you replace the Hodgkin-Huxley equations with this discrete time map for each class of neurons. And I haven't discussed how you connect them together in the network.
Okay.
That's through the synaptic connections or gap junction connections I mentioned at the beginning. But for every class of neuron, you just as you would develop a Hodgkin-Huxley neuron, for that class of neuron, you make what we call data-driven forecasting, a DDF neuron for that class of neuron. Okay.
And you decide how many classes they are. You don't learn that from the data.
Oh no, you have to learn that from experiments on the network that you're trying to characterize. This does not replace experiments at all. It rests on experiments, but focuses on the thing you need to know to put the biological information in the experiments into a network that would, you hope, explain the functionality of the network. So good questions. Any others before I go on?
I thought the question here, Henry, was a dimension of U, how many components does the vector have? And what does it mean in terms of how many bases do you need to really faithfully reconstruct that function?
Are you asking the dimension of
the vector U? Yes.
The vector U has the dimensions of however many things about whatever system you're talking about, you can measure.
So it could be millions, or it could be hundreds, or
for individual neuron in current clamp experiments, the dimension is 1. It's
okay, sure. Yeah.
Okay. If it were the atmosphere, Gert, you could measure lots of things. All right, but I'm not ready for that yet. Okay. All right, here's how you do it. Here is our map. It's the components of the vector. I'm still talking about a d-dimensional system so that you get the generality of what this is. And the measured quantity at time n+1 is equal to the measured quantity at time n plus this combination of functions. And if we choose one of these functions, I'll tell you which ones we in fact use. The determination of the weights of the various functions, which tells you how important are the sampled locations on the distribution in the U space, is a linear algebra problem. And if you write the problem as a matrix problem, then I call Y the difference between the observed quantity at time n+1 minus the observed quantity at time n. You know these because that's your data. I call the radial basis function, which depends upon n and q. The q's label the centers that you chose, and the u of n tell you about the data that you didn't use as a center. And I call that matrix Z. And what you have to do is invert Z, multiply it by Y, and that gives you the weights. Once you've determined the weights, this rule is your map that allows you to project forward in time past your observation time of whatever it is that you observe. All right, and how do you do this? Well, the matrix is rectangular, and therefore, in order to define an inverse, you have to provide a regularization procedure. The regularization procedure is known as Tikhonov regularization in some circles, and it's known as ridge regression in others. But it's standard, and this is not anything that we invented. And here's the answer. If you know this matrix Z because you know this function and you know the data, and this is the expression you get for the weights in that representation of the vector field for the map. All right, so the parameters up to this point are the weights, the number of centers you choose, and I'll have a little bit to say about that, the distance in the space of measured variables you want to affect your interpolation, and this regularization parameter. It's called the Tikhonov regularization parameter. And as I've said, once you know it, then this is your predictor. So let's go on. There are many, many choices for good functions of these radial basis functions. Here's one. It's a Gaussian. It says that if you are at a point U in the space of the quantities you can measure, then the centers, the samples that you take from the data that influence where you are, is essentially 1 over the square root of R, or the RMS variation. Here it is. This is 1 over twice the variance in usual notation. But that's not the only radial basis function. There's another one that was actually developed in people studying crops in agricultural fields, and they wanted to know from what they measured how they could extrapolate or interpolate to the crops in the areas they didn't measure. This one is called a multi-quadric. I don't know why. And it has the same kind of feature. The parameter sigma that tells you over what range in the measurement space your samples are important enters this way. There are whole lists of various radial basis functions. We've tried a few of them, and the truth is they all seem to work more or less the same way with the data that we've worked with. And so I'm only going to report on using the Gaussian. If you want to know what happens when you use this, I can tell you, but it's not much of a different story than using the Gaussian. Whoops.
Does your forecasting explicitly use the Hodgkin-Huxley equations, or
No, it doesn't.
Right.
Right now, I don't have any forecasting at all. I've lost the slides. Let's see if this works.
You did have 58, which seemed like a lot. But
oh, well, that's because there are 2 talks here.
Ah.
And lots of pictures, except I don't have any of the slides or any of the pictures anywhere. I don't know where they went. So if you pardon me, I'm going to stop sharing and see if I can find out what happened to my document.
If you can successfully multitask, it'd be nice to hear a little bit more about what the neuroscience data set. More explicitly, what this network looks like and what dimensions it has. So you have
I have that explicitly.
Okay.
For, learning about the voltage forecasting from neurobiological data. I just don't know where. I don't know what happened in my talk. Hang on. Okay, for some reason, I'm stuck.
It was Acrobat, right?
Pardon me?
It was Acrobat.
Yes, but this particular computer doesn't have Acrobat on it. Hang on a second. I'll be right back. I'm going to log into the this with another laptop. Hold on. Sorry about that.
How robust is the choice of the number of centers that you assume at the beginning? Is there a way, an iterative way to correct that, or is
What we find is the following. I'm sorry. I'm going to still look for the talk here somewhere. Clearly, one center isn't enough to sample the distribution. And so we have done tests on what I'm going to show you with a few hundred centers and a few thousand centers. And what you find is that the prediction error becomes independent of the number of centers after you get more than a few hundred. Sometimes it's 100, sometimes it's 200, sometimes 500, but it's only a few, and that's not a big deal.
Shouldn't the number of centers depend on the number of neurons you're simulating? And if so, how many?
No, the number of centers is for each neuron. We're not simulating the entire network with this. We're simulating each neuron the same way as you would building a Hodgkin-Huxley model for each neuron, and then putting those, what we call DDF, data-driven forecasting neurons, into your network. But I can't put them in if I can't give the talk. Hang on a second, I'm converging here. This time, and
recording in progress.
okay. I have to now.
We have some echo now, though.
Yeah.
You have to turn one sound off.
Yeah, no, I have to. There we go. Is that okay?
You may have to mute the other computer or so. Yeah.
Exactly. All right.
yeah, if it's easier for you, you can just share the PDF with me. Like,
Okay, what I'm going to do is mail it to you. Okay.
yeah, okay, no problem.
I-
I mean, we can see it now, but if you prefer, like,
Oh, can you see it now?
yeah, I can see it now.
Oh, excellent.
We
Sorry.
saw it earlier too. Yes. Yeah.
That's okay, no worries.
All right.
But could you do full screen, please?
Gert, I hope so.
Control L. Yeah. It's good. Thank you.
That, okay. So where were we? I really apologize. I came to my office on Monday for the first time in 2 years, and nothing worked very well. Okay, here's where we were. And I'm sorry, did I answer? The question was how many centers you need? And I'll show you not the calculations we did, but I'll repeat the results. And all of the results that I'm going to show you use this Gaussian radial basis function. But again, the other of which this is an example are equally effective. Okay, I'm going to do three things here. I'm going to go to the simple sodium, potassium, and leak model of Hodgkin-Huxley. I'm going to generate data for the voltage and all the gating variables and pretend that those are my data. This is not the realistic situation in a current clamp experiment, but it shows you how the method works. Then on that same simple model, I'll take only the voltage data as the measured quantity, and I'll build a DDF neuron with those voltage data alone. And that will require a pretty well-known method from nonlinear dynamics that I'll remind you of. Then I'm going to examine experimental data on current clamp experiments on HVC, which is the nucleus in the avian song system that whose network I'm aiming to build. The data came from the Margoliouth lab at the University of Chicago, collected by Dan Maliza when he was a postdoc there. Okay, so this is the detailed model again. Sodium currents, potassium currents, leak currents, and here's a stimulating current, which is entirely up to the user. I'm not going to discuss that in any detail. I'll show you what they are. And here are the familiar first order kinetic equations and so forth for the gating variables. I'm not going to discuss how you solve the Hodgkin-Huxley model. You probably all know that. It's important as a step, preliminary step, to write the general structure of these Hodgkin-Huxley models. It's C dV dt, which is a current, the rate of change of the charge on the capacitor represented by the cell membrane. There's intrinsic dynamics, and the stimulating current is additive. The gating variables depend upon the voltage, but not the stimulating current. And what we want to do is integrate this equation over one time step. So we integrate it from time n to time n plus 1. And that's what I'll show you here. And when we do that, we get the discrete time map for the voltage, which involves the intrinsic dynamics, which we're going to represent by one of these functions, because in principle, if I give you only the data, I do not know the Hodgkin-Huxley model, and I don't need to. And the other is the integration over the stimulating current over one time step. But we know the stimulating current because we have to specify it. So as I said, the first thing I'm going to do is assume that you measure the voltage and the gating variables. So we need two kinds of maps, one for the voltage itself and the other for the gating variables. This is just a technical point that's not important. So here's what happens in that scenario, which I emphasize is not the realistic protocol of a current clamp experiment, but we're seeing if the method works at all. This is the stimulating current that was chosen. And this is a graphic with the data that we generated. We then trained the DDF neuron, the data-driven forecasting neuron, on the data from the voltage and the gating variables, and then we forecast the voltage. And you can see in red the forecast, and you can see in black the data that we worked with. So it works in this unrealistic case, but now we want to go on to the real case, which you only observe the voltage. And here's how one thinks about that in nonlinear dynamics. This will be familiar to many of you. The voltage is a projection onto one of the axes in the state space of the system, which in the simple NAKL model, this Hodgkin-Huxley model, consists, which is four-dimensional. So in order to know in which space we should choose those centers and do the interpolation, we have to effectively unproject the data. And that's done through the method of time delays. And what we do is we create a vector out of the voltages and its time delays. Here we have d sub e time delays, and the time delay is a number that we have to select and the size of this vector. The physics of using the time delay is important to remember, because it isn't that the voltage, we're predicting the voltage only. That's the only thing we can forecast, because that's all that was measured. But the other variables in moving from some time n minus that time delay to the time you have in mind is influenced in the actual system by all the variables in the system, in our case, the gating variables. So what we need to do is finally build a discrete time map, which is this form for the voltage only. So it's the voltage time n plus 1 equals the voltage plus the integral of the stimulating current, which we know. And this is a function now in this time delay space. I'm assuming that many of you have seen this many times. And again, it's not something that we invented. But here is what happens when you take this sodium, potassium, and leak Hodgkin-Huxley model. You generate data with it by solving the differential equations. And you train it on some stimulating current. Once you've trained it, you can present it with a new stimulating current. And here's the result of the data from solving the underlying equations. And here's the forecast when you have only observed the voltage using the method that I described. And you can see it's remarkably good. It's not perfectly accurate in the subthreshold level, but in this particular example, it's captured all of the spikes and therefore is as good as the Hodgkin-Huxley model that generated the data. I'm going to pause a second because it's very important here. In this numerical example, we generated the data by hand. In the next set of slides, I'll show you the data was measured in the laboratory. We do not know in the laboratory examples the detailed Hodgkin-Huxley model. We have guesses for it, but using this method that I'm describing, we don't need to know all of that. What we do is build forecasts of this kind on the voltage observations only.
What's the minimum amount of data you need before the prediction degrades? You use 500 milliseconds there. Like how smaller can you go? Like 100 milliseconds, 10 milliseconds?
On the training?
Yeah.
Oh, it really depends upon the stimulating current. All of this, I'm sorry. The point here is that you choose a stimulating current and you train the coefficients in this radial basis function representation. I don't know a mathematical answer to your question, but the biophysical answer is that the stimulating current should drive the actual neuron in both depolarizing regions, hyperpolarizing regions over a number of spikes. The typical number that we use is 10 or more, and that really depends on the stimulating current.
Okay.
What you're trying to do in a mathematical sense is have enough samples that sample the orbits of the underlying dynamical system. Here, the individual neuron that you're stimulating with the current samples that distribution in that space pretty well. All right, and that's the best I can give you. I don't have a mathematical answer.
Okay, thank you.
All right,
Professor,
I'm sorry, go ahead.
for the centers that you choose for this model, do you choose every single data point
No,
as a center? And if not, then how do you choose which ones become centers and which ones are discarded?
what we do is use what's called k-means, which you probably know, and we choose more centers where there's more variation in the observed data and fewer centers when there's no particular observation. So in the case of voltage, if the voltage is more or less constant, we don't need a lot of centers because nothing's happening. But when the derivative of the voltage is large, namely at a spike, we need more centers to track that. And as I said, we've done that with thousands of centers, and we've done it with just a few hundred. And in these problems, there's no difference really. Okay.
Yes, makes sense.
All right. All right, here's real data. And as I said, this was collected by Dan Melisa when he was a postdoc in Chicago. Here's the stimulating current. This was designed by Dan Melisa and one of my former students, Mark Kostick, when he was a PhD student here. We sent Mark off in probably the spring of some year to work with Dan, and they devised a number of currents that do the job of driving the neurons in the laboratory, both over, and here's the voltage response, over hyperpolarizing spiking regions and threshold regions, so that the space of response of the actual neuron is well sampled. We took these data and took this form of the map and made a predictive neuron, and here's the prediction. Here's the stimulating current in the region of prediction. Again, this is 500 milliseconds of prediction, and I'm pretty sure here we used, again, 500 milliseconds of training, and the prediction here is the following. The prediction is blue of the voltage, and the data itself is in the black. Again, it does extremely well on the spikes, pretty impressively well in the region of hyperpolarization, and occasionally it misses a spike. If it didn't do that, it would be too good to show. So now that's cool. We did one more thing. The data was collected in the lab by putting an electrode through the, or patching on to the cell wall of an individual neuron. And the data was collected in epochs of time, order 3 to 6 seconds. And then the neuron was allowed to rest for a while, and with a different waveform for the stimulating current, another collection of data was given. What I'm going to show you is that if we use data from one epoch with one waveform, that's our model of the neuron, that we can predict what happens in another epoch with a different waveform. So these are the data in epoch 25 for that particular neuron. And this is the result of the experiment with a different stimulating current and a different voltage response, of course. So to say it again, we're going to build our DDF neuron on data from this epoch and predict what happens with this different current in another epoch. And I'll jump to that because I've spent all this time trying to reconnect here. Here is the epoch 26. There's a selection window here. There's 1,000 milliseconds of stimulating current and 1,000 milliseconds of data. And you can see that the neuron that was trained on a different stimulating current is behaving quite remarkably like the data on this stimulating current. It actually was good that it didn't do well on these square pulses, and there's a physics reason for that. These square pulses have very high frequencies as they go up and down very sharply in time. And those high frequencies do not get into the neuron through the RC time constant of the membrane. And so the neuron doesn't actually know about them. So it's not a surprise that it doesn't learn very much about them. And my recommendation is to get rid of these square pulses altogether, even though they're very popular in the neuroscience world. That's just an ad for this. All right, we did another kind of experiment. I'll say it in words, and then because of the hour, I'm going to go to the conclusion. What we did is we built a tiny network. I said that you could use these DDF neurons in the place of Hodgkin-Huxley neurons. I say it again, the DDF neurons know all about the biology because the voltage knows all about the biology. It comes from all of the operations of all of those gating variables and the biochemical concentrations, typically of calcium or whatever in the cell. So we built a 2-neuron network. We figured if this didn't work, we were sunk. And we put Hodgkin-Huxley neurons at the nodes of the network. We stimulated it with a current that we chose, and we recorded what happened in neuron 1 and neuron 2. And then we replaced the Hodgkin-Huxley neurons with these DDF neurons and did the same experiment. And here's the result of that. This is neuron 1, and this is neuron 2 in that network. Again, black are the data with Hodgkin-Huxley neurons. Red are the results with these DDF neurons, which we trained using the Hodgkin-Huxley neurons. This tells you that you can do circuits. We haven't built big circuits yet, but that's on the plate for future. I have one more thing to say, and then I would be happy to entertain more questions about details or the philosophy or whatever. Why is it that the currents in the Hodgkin-Huxley model are additive? This was a puzzle to us because it means that if you build a Hodgkin-Huxley model, you don't have to learn anything based on the stimulating current. If you give me the stimulating current, it tells you everything. And the reason why it's additive, which makes the form of these DDF, just dynamical maps, very simple, is that in all conservation laws, they're additive. For example, in fluid dynamics, the dynamical variables are velocity, mass densities, temperature for energy, and each one of them has the form of the rate of change of the appropriate dynamical quantities equal the sources. This is F equals ma. This is energy change, and this is mass change, if any. If there are no sources or sinks of mass, the right-hand side is 0 here, but the forces enter here linearly as well. So the tool that we used, and I'll go back to it, and that's where I'll stop, which is this. Equation 15 is the critical tool. It says you train this on one stimulating current, you learn that integral over the intrinsic properties, and you can predict for another stimulating current, because this is the only way it enters the model. Okay, that's it. And then I have a slide which says we have a moment of rest. So I would be happy to address any questions you have, and I apologize again for the the flakiness of the Wi-Fi connections. So are there any questions?
I was curious about how long it takes to train these things. Is it quickly?
The training well, it depends on your computer and so forth, but it's extremely fast to train them. The more centers that you use does not influence the training very much, but in the operation of the model, the more centers you have, the slower the predictions are. So you want some numbers for the, we took a model that we had built, a Hodgkin-Huxley model that we built on that data I showed you from Dan Meliza. It had 14 state variables, voltage, gating variables, calcium concentration, conservation equations. And we built a model on just the voltage data that I showed you. And that model runs in 500 milliseconds or 1,000 milliseconds in 10 seconds on a laptop computer. There was somebody who had a question in the chat. Could you say that question?
Yes, my question was, does the model sort of drift over longer simulations? Like, do you expect to see any accumulating error terms?
Well, in principle, if the intrinsic properties of the neuron don't change over time, the model is not going to change over time. If the environment in which the neuron finds itself changes, then both a DDF neuron and a Hodgkin-Huxley neuron are going to see the same drift because of the environment. And that's a feature of any neuron model put into an environment.
I was wondering about that. If you have any plasticity effects, so you would have to relearn the DDF neuron, like having a new centers and relearn the weights.
No,
right?
the plasticity takes place at the synapses. And in order to build the network model, individual neurons aren't going to learn anything. You may change the AMPA conductance, for example, but that would be part of your data set. But the connections among neurons, which are determined, as I pointed out, by the voltage alone, if you have plastic synaptic connections, you will learn that as you build the network once and for all. And then the only thing that the network wants to know is, what is the external stimulation? In the case of the birdsong system, it's auditory stimulation of the network as a whole. But you don't have to relearn the neuron dynamics. That's
And
fixed.
what about homeostatic plasticity, where you have some channels of the neuron that change, not the synapse, but like other channels in the neuron that will...
Oh, if the intrinsic properties of the neuron change, how would you know that if you were building Hodgkin-Huxley models? You would have to change the neuron at every node where there was a change. And this DDF neuron is not no smarter or no dumber than a Hodgkin-Huxley model, just much more efficient in computation.
But if the simulation are that fast, you could include like those things in the network simulation that you're doing and kind of relearn perhaps the DDF stuff if when needed.
Well, but again, the idea is that you would like to build a network with biologically based neurons. The DDF neurons are based on biological data. The way a network learns is that the synaptic connections get stronger or weaker and build the network based on it, however it gets trained from external stimuli. And it's no different here. It's just that the Hodgkin-Huxley neurons predict the voltage and all of the other variables, state variables, the gating variables, and so forth, that you never need for the dynamical connections. And therefore, these DDF neurons, which are the reduced models, provide you with computational efficiency that you can use to build bigger networks, much bigger networks.
Can you write the DDF as an explicit dynamical system? I mean, like, can you write it as differential equations? Because you have this basis function you're expanding, and can you compress it down into like a more simple differential equation at each point that you would be obviously harder to calculate because it's you're now having to integrate them as opposed to.
The answer is probably no.
Okay.
And that's because the DDF is formulated as a discrete time map going from some time to the next time plus that time plus delta t and so forth. In order to write that as a differential equation, you'd have to take delta t equals 0.
Mhm.
But the DDF is built on observations, and you don't have delta t equals 0 in the observations. The predictive power is exactly the same as the underlying differential equations, which you ascribe to the neurons themselves.
Okay, but the DDF would be different if you had different time steps of your data, basically.
If you sample your data every hour instead of every tenth of a millisecond, the DDF will be completely wrong. So you have to sample your data frequently enough to catch the variation.
Oh yeah, but what I'm saying is if it's like one millisecond versus 2 milliseconds or whatever, the DDFs would be completely different for those two sampling frequencies.
Yes, in principle they would. But again, so what is the actual delta t that was used in the experiment? I didn't mention that. It was 0.02 milliseconds, which is a sampling rate of 50 kilohertz. If you sample the neuron dynamics of the neurons that were actually observed in the lab at 25 kilohertz, there's no big deal. If you sample them at 1 hertz, you lose everything because it averages over the spikes.
Okay.
So you have to get data that has been sampled frequently enough. Collecting data at 50 kilohertz sampling is no big deal. All right. And if you think that's too frequently, you can throw out half the data points.
Right.
Okay. And that's okay. But if you sample it too infrequently, you miss the details of the sub-threshold behavior, as well as some aspects of the spiking. And the sub-threshold behavior is part of the biological dynamics that it's important to incorporate. At 50 kilohertz, it's fine. You don't need 150 kilohertz. That's too frequent, and then you would throw out data points. But that's a really good point.
Thanks.
Sure.
Did you compare the performance of this method with other forecasting methods? For example, if you would use artificial neural network or something else, having my comparison?
No, and I'm not planning to, because artificial neural networks are simply, well, it depends on what kind you're talking about. If you're talking about a feed-forward network, that's essentially the same as the data assimilation techniques that are used in the work that we've done with the Chicago lab over many years now. It is mathematically the same, except it doesn't have a biological interpretation. But you could, in fact, use it to learn the function voltage to voltage. And if you use recurrent neural network, you have to be careful of how you build that and train it on the data. You don't have to address those questions in this methodology. It works on the data alone. And I'm not quite sure if I'm answering your question. Do you want to know if it works faster than a feed-forward network?
Or if the predictions are better?
I have no idea. How much better than the predictions I showed you do you want to have?
No, for example, you could have better predictions with your method than a relatively small neural network, something like that. I was pointing in that direction.
Okay, let me not be so dismissive of using artificial neural network. The representation of the vector field can be considered a neural network, but the things that are at the nodes of the computational network are these radial basis functions, which are designed to interpolate among the data that you have. The training of them is linear, and this is not something that we invented. It's a well-known method in applied math, as I said, for many years, since the 1980s and 90s. So you could do it with other methods.
How does it compare with the data assimilation method?
Data assimilation requires two things: data, and that we have. It requires a detailed model that you prescribe based on your sense of what ion channels are there and of the kinetics of all of the gating variables. So the model that I described that we built on data again collected in the Chicago lab. And in fact, I think Dan Melisa was one of the co-authors on this paper, had 14 state variables and 70 parameters. And the computations that are required to estimate all of the state variables that you don't observe, namely all of them except voltage, and all the parameters, maximal conductances, time constants, and so forth, all of the ones that go in even to the simplest equation, is quite long. So the training of these networks, again, on the data alone, these networks, these model neurons that predict only the voltage, is very rapid compared to data assimilation. And it doesn't need all those details, and it can't tell you about them either.
And so like there are 70 parameters in the differential equations, and there's 100 parameters in your basis functions.
Yes, but
1,000. I'm just trying to get the scale.
All right, there are hundreds of parameters, but they enter in nonlinear ways in the detailed Hodgkin-Huxley models. They're the time constants in the kinetic equation. They're
No, I'm talking about the radial basis functions. You have So you have like 10 radial basis functions and then 1,000 parameters in your Hodgkin-Huxley equation.
Well, it's more like there are 100 weights that enter the interpolating functions. And in the Hodgkin-Huxley model, but they enter in a linear fashion, so you can use the ridge regression to do the calculation very fast.
Yes.
And that's really the competitive advantage. The other being what I've said several times is that you only predict what you observe. And but you didn't throw out any of the biology that's there in those time delays. Or biophysics. Let me pretend that I'm a physicist here. Okay.
Hi, Henry. Thanks for inviting me. It was fun to see that data being used. I had a question about the centers. I apologize if I missed this, but are they just in a grid over the state space, or is there some way that you're focusing your attention on regions of the vector field where the derivative is higher?
It's the latter, but it turns out that that's kind of a bells and whistles on the general method. We did it both ways. And if you sample only the dynamics in the subthreshold region, you need fewer centers because the derivatives are small. But if you want to sample the spikes well, you need a few more. So the method that we use essentially puts more centers on the spikes than it does in the subthreshold region. But the truth is, if we sampled totally uniformly, it didn't much matter. I admit the method that we devised was cooler, but that's all I can say about it. It's not better.
Did the centers move around during the training, or you just
No. Those are your observed data. Those are your voltage data along with its time delays. They're fixed. You give us the data as you did. We put them in a library and we never touch them. We just use them and say you're sampling the distribution in the state space where the neuron is operating. And you use this method to give you an interpolating function among your data. You don't extrapolate beyond it because that's always a bad idea.
Thanks.
Sure. Good question.
We have time for more questions, so please don't be shy.
Hi, I had another question about just scaling and what you think that these kinds of models would be best used for.
Oh, that's a crystal ball question, right? Are you talking about scaling a network? I'm not sure what you mean.
Right. I realized that was pretty vague. I'm talking about scaling in terms of maybe trying to build exactly like you said, a network, but that would require then having simultaneous voltage recordings from multiple neurons. That's right?
No, you would need that in order to have an experimental basis for building the network. That's correct. And that kind of data didn't exist when these individual neuron data were collected. But now using calcium indicators in fluorescence and 2-photon microscopes for localization, people have just been able to start collecting the experimental information on the structure of the network. And we intend to use every bit, literally, of those data in building up a network.
But you need to know the input, right?
You need,
in order to make your map between the input and the output, which is what you do at each point, you need to have a handle on what the input to the neuron is, right?
well, let's go back and talk about individual neurons before we put them in a network, all right? Individual neurons, you can specify the stimulating current any way that you want. Some ways are better, some ways are not so good. And that gives you the intrinsic properties of this input-output map. That is the correct way to characterize it. When you're done with the experiments on the individual classes of neurons, then once you put it into the network, how does a neuron get stimulated? It gets stimulated by something external to the network, acoustics in the case of songbirds. It comes from the auditory system. But it also gets stimulated by the dynamics of all of the other neurons in the network. And that, of course, tells you how the network works. So you don't need to specify anything except what you specify when you study how a brain, whether it's in the avian system or a mammalian system, works when you stimulate it. Does that answer your question if I understood it correctly?
I think so.
Okay. If you think about it and you don't think so anymore, send me an email. I'll be happy to talk to you. Or if it takes you a few weeks, maybe we can talk in person. Who knows? Okay, Frederick,
I mean,
thank you very much.
What you're presuming is that
Oh.
what you're presuming is that you'll be able to assign identities to every neuron, and it'll come from a class, and you'll learn via voltage clamp. I mean,
Current clamp.
Current clamp. How each class works, and then that'll be a piece that you add in.
Yes, and that's exactly what you
Yeah.
do with the Hodgkin-Huxley models.
Yeah,
Exactly.
yeah. Okay, that's why we spent so much time talking about it. Now I understand why you led with that. Okay.
Right. Well, and to repeat what our experience is, if your Hodgkin-Huxley model has 10 gating variables,
Mhm.
what we see is an improvement in the computational speed of just forecasting voltage of order 10.
Right, because you don't have to integrate all those equations.
Exactly, but the biological information is still encoded in those coefficients, just not
Mhm.
in maximal conductances and time constants of the
Mhm.
kinetics and so forth.
Yeah, there's a solution to the differential equation, and you're approximating that, even though we don't know what it is.
Exactly right. And we know that it is some function of the state of the system, and that was the technique that we used to do that. But quite frankly, it took a while to swallow this because it's not a detailed biophysical model.
No,
And
but it also is. It's just you're washing out the model basically. There's no model. It's you've gotten rid of all the parameters and then there's just it's a black
That's right.
box.
They're still parameters. They just don't have immediate biophysical significance. Now, I have an idea on how we can use them anyway, but for biophysical questions, but
That sounds interesting.
we haven't done anything that I could tell you about. If you want to hear the ideas, we can take another couple hours.
Sounds fun. Maybe I'll shoot you an email.
Okay. All right, Frederick, thank you very much.
Thank you, Henry.
I'm so sorry for the defects in what was left of my office in the last couple of years,
But that's okay, don't worry.
we're fixing them. Okay, take care, everybody. Bye-bye.
Bye-bye.